Hello and welcome to the next class of human embryology. Today's session will be development of liver. Today's session will be development of liver. Today we shall be discussing about development of liver, the bile duct, the pancreas. We'll discuss how the respiratory system is formed, how the spleen is formed. We'll also discuss how the diaphragm is formed. All right. We'll discuss all these points: the liver, the biliary apparatus, the pancreas, spleen, the respiratory system, and diaphragm, right? So highlights of today's class are as follows. The summary of today's class is as follows. First, we'll discuss liver and biliary apparatus. The liver develops from what is known as hepatic bud. The liver is very unique to develop from hepatic bud, which arises from an outgrowth of ventral wall of the foregut. So this is the foregut, and say suppose this is the midgut. So at the end portion of the foregut, somewhere here, a small outpouching develops. We call it hepatic bud. Hepatic bud at the terminal portion of the foregut. Whereas the pancreas, it develops from two buds. Pancreas develops from two buds. The pancreas develops from a ventral bud and a dorsal bud. A dorsal bud and a ventral bud. So ventral pancreatic bud, dorsal pancreatic bud. These two buds will undergo fusion at the junction of foregut and midgut. These two will undergo fusion. So there are two buds. The two buds will undergo fusion, all right, by rotation of the gut. At the time of rotation of the gut, and the ventral bud forms the lower part and head, whereas the dorsal bud forms most of the pancreas. All right, dorsal bud forms most of the pancreas. Next, we'll discuss about the spleen. Spleen develops from small spicules, which are derived from the mesoderm. Mesoderm, we call it dorsal mesogastrium or dorsal mesoderm. Next, we'll talk about the respiratory system today. To summarize, respiratory system. The respiratory system will be forming from the endodermal derivative of the foregut. Endodermal derivatives of the foregut. The caudal end of the respiratory system, the caudal end of the diverticulum, a small diverticulum called as endodermal diverticulum, is giving rise to respiratory system. So this respiratory system is formed by the endodermal diverticulum, which is derived from the foregut. The proximal portion of this endodermal diverticulum gives rise to the larynx and the trachea. The distal portion of the diverticulum forms the right and left lung buds. Right and left lung buds, and which develop into right and left lung respectively. Right and left lung respectively. Around the lungs we have got pleural cavity. Around the heart we have got pericardial cavity. In the abdomen we have got peritoneal cavity. All these three are derived from, last time I discussed in my previous class, it is derived from the intraembryonic mesoderm. The intraembryonic mesoderm gives rise to the pleural cavity, the pericardial cavity, and the peritoneal cavity. This point, please make a note. You tend to forget this point. It is the intraembryonic mesoderm. So next is the pericardial cavity is derived from the mid portion of the intraembryonic mesoderm. The peritoneal cavity is formed from the right and left limbs of the intraembryonic mesoderm. The pleural cavity is formed by the right and left pleuropericardial canals of the intraembryonic mesoderm. So pleuropericardial canal gives rise to the pleural cavity. The pericardial space develops into the pericardial cavity from the mid portion of the intraembryonic mesoderm, whereas the caudal portion of the intraembryonic mesoderm, wherein the right and left limbs of the intraembryonic mesoderm are responsible for the formation of the peritoneal cavity. Next, we talk about, in the last part, we will talk about the diaphragm, the main chief muscle of respiration. This is formed by small parts or portions in relation to what is known as the septum transversum. All right, and it is very unique that these small portions will join to form a large muscle. The somites will join to form the large muscle called as diaphragm, the major muscle for respiration. So let us begin our discussion proper. We start our discussion with liver and the biliary apparatus. The liver, as we all know, it is the largest organ of the body. In an adult life, the liver is around 1 to 1.5 kgs in weight. It is a very largest organ, in fact, largest solid organ of the body. The liver has got various functions. It has got endocrine function, exocrine function, hematopoietic function, metabolic function, biochemical function, and apart from this, immunological function, helps in phagocytosis, develops various antibodies, develops immunoglobulins. It is a factory of formation of amino acids, from amino acids, proteins. It is factory of metabolism of fats. It is a factory of metabolism of the glucose and various other metabolites. So liver is a very active and complex physiological organ. The formation of liver is at the level of terminal portion of the foregut. We all know that terminal portion of the foregut gives rise to the second part of the duodenum, upper half of the second part of the duodenum. So the terminal portion of the foregut is responsible for a small growth is formed. We call it hepatic bud. This hepatic bud consists of rapidly proliferating endodermal cells, rapidly progressing or rapidly proliferating endodermal cells. The rapidly proliferating endodermal cells, which can grow above and below. So they grow above to form ventral mesogastrium, and it also gives rise to part of septum transversum. The ventral mesogastrium and the septum through into the septum transversum. This septum transversum is actually a part of intraembryonic mesoderm, which is also growing along with the ventral mesogastrium. Now the hepatic bud starts dividing into it undergoes small divisions into pars hepatica and pars cystica. Pars hepatica is a large part and pars cystica is a smaller part. The pars hepatica forms the liver, whereas pars cystica gives rise to the gallbladder. It gives rise to the gallbladder and the cystic duct. Alright, the pars hepatica again divides into right and left branches, right and left small branches. Give me a second. Yes. So the pars hepatica divides into two branches, right and left branches, which gives rise to right and left lobes of the liver. It gives rise to right and left lobe of the liver. In the third week of intrauterine life, please remember, in the third week of intrauterine life, the weight of the liver is 1/10 of total body weight. 1/10 of total body weight. So it is occupying almost the entire abdominal cavity in the third week of intrauterine life. Whereas in the seventh week of intrauterine life, third month of, not week, third month of intrauterine life, I'll write it here. So in the third month of intrauterine life, the liver contributes to around 1/10 of the body weight, total body weight. So it occupies almost entire portion of the abdomen, upper abdomen. Whereas in the seventh month, it is reduced to 1/5 of the total body weight. All right, 1/5 of the total body weight. So relatively, the body is growing faster. So what happens here is the terminal portion of the right and left branches of the pars hepatica, they divide to form what is known as the hepatocytes. Hepatocytes are the functional unit of the anatomical unit of the liver. Hepatocytes. These are cuboidal cells, hexagonal in nature. They are small cuboidal cells, and they are arranged in plates, clusters. They are arranged in a form of layers. Okay, we call it laminae. All right. So in histology, I'll discuss how beautifully a hexagonal hepatic lobule looks like. But here, please remember, each of these hepatocytes, they are clumped together to form what is a hepatic trabeculae. The hepatic trabeculae. These hepatic trabeculae, they consist of vitelline and umbilical vein that runs through it. And we have got vitelline blood vessels and umbilical blood vessels, which form what is known as, they communicate with trabeculae to form what is known as an important structure called as hepatic sinusoids. Hepatic sinusoids. The hepatic ducts are branches which are repeatedly canalized and to form biliary passages. So what happens is these hepatic trabeculae, they differentiate into parenchyma. These hepatic trabeculae, the liver cells, the intrahepatic biliary system. So the hepatic trabeculae, are nothing but they are formed by the hepatocytes on one side, and also the intrahepatic biliary system, biliary duct system, biliary duct passages, small passages or systems. All right. Apart from this, we have got certain immunologically significant cells of the liver called as Kupffer cells. Kupffer cells. Then we also have the interstellate cells, the stellate cells. We have got the connective tissue cells or hematopoietic cells. The stellate cells, the hematopoietic cells, all these are very, very important. All these are most important structures which are, along with hepatocytes, which are intermingled. These structures are basically contributed and supported by septum transversum. Septum transversum is a part of intraembryonic coelom. Just now I told you. So these are contributed by septum transversum. So what happens is the hepatic bud divides into right and left branches. That is a pars hepatica and a pars cystica. Pars hepatica divides into right and left branches again. the giving rise to the respective lobes of the liver. They divide and redivide to form hepatic trabeculae. These hepatic trabeculae nothing but they are combined in intrahepatic biliary system and also the hepatocytes. Just I'm revising, all right. And the blood vessels which open around these form what is the hepatic sinusoids, and in between these cells we have got specialized cells contributed by the septum transversum. So this reorganization of entire hepatic system gives rise to what is known as a functional component of the liver, so that the normal functions are carried out. So what happens is the hepatic bud and the mesenchymal cells. See, blood vessels derived from the mesenchyme, is it not? So the blood vessels and the hepatic bud duct, both are forming, reorganization is occurring. They form what is known as hepatic lobule. The central portion of this lobule, we have got central vein. All right, we have got central vein. So multiple hepatic lobules like this are arranged. I'll teach this in anatomy and histology proper. But this hepatic lobule will have bile canaliculi, the portal triad, and sinusoids of liver. They have got bile canaliculi, the portal triad. So portal triads arranged like this, and bile canaliculus, the hepatic sinusoids, the central vein or hepatic vein, the branch of the hepatic vein in the center, right? The formation of peritoneal folds in the liver from the septum transversum helps us to form attachments to the lesser omentum and greater omentum. So the ligaments of the liver are formed. We have got falciform ligament of the liver. We have got coronary ligament of the liver. Then we have got triangular ligament of the liver. All these are connective tissue components of the liver. These components are nothing but the portion of the lesser omentum. They form what is known as the mesentery. Gives attachment to the ventral mesentery or ventral mesogastrium. Ventral body wall, we call it ventral mesogastrium. Hope you have understood this. So the fetal liver, please remember the important functions of fetal liver are hematopoiesis. So for the first 3 months, hematopoiesis occurs in the liver. Second important function is bile secretion. Bile secretion starts 12th week of intrauterine life. The bile secretion starts at 12th week or 3rd month, the end of 3rd month. Is not the begins at 12th week. At around 12th week of intrauterine life, the bile is released into the foregut in the second part of the duodenum and through the intestine. The first stool is passed. The first stool passed is called as meconium. The first stool passed is the meconium, and this is what helps us to understand. So if I draw all of these with the help of a diagram like this, so this is the liver, all right, this is the liver, and we have got the right and left umbilical vein and right and left vitelline vein which enter into the liver. So we have got, say suppose this is the umbilical vein, and this is the left sided umbilical vein. All right, this is the right vitelline vein. This is the left vitelline vein. All right, this is the demarcation between the right lobe and the left lobe. All right, and here somewhere we have got the sinus venosus. This sinus venosus is draining into the heart, is it not? This sinus venosus is joined by the branches from the right and left side of the liver, right and left lobes of the liver. All right, here at this point of time we have got right common cardiac vein and left common cardiac vein which joins the sinus venosus like this. So we have got right common cardiac vein, we have got left common cardiac vein. This is the sinus venosus, which is draining into the heart, all right. This derives from the branches of the sinus venosus. Right and left branches are two major branches which carry blood from the liver into the sinus venosus. In future these are going to form the hepatic veins which are entering into the inferior vena cava, the hepatic vein which is draining into the inferior vena cava. The right and left common veins are attached to the sinus venosus at this juncture. All right, so this is how the hepatic sinusoids, so there is a communication between the hepatic sinusoids in between the umbilical vein, so these are the umbilical veins. I'll label it left umbilical vein, left vitelline vein. This is the right vitelline vein, and this is the right umbilical vein. So this is a demarcation between the right and left liver, and this is the mesentery attachment, yellow color. Hope you have understood the segmental changes here. We'll also discuss how I told you that these hepatocytes are arranged. So these hepatocytes forming the hepatic trabeculum, they are linearly arranged small cells, I told you, is it not? So the cells are something like this. So hepatocytes, small cuboidal cells, each of these cells are linearly arranged like a plate, laminae. Alright, we have got multiple layers like this. I'll draw two layers and show how these layers of hepatocytes are formed. So they form a part of hepatic trabeculum. In the central portion, if you see, in the central portion, if you are going to see, in the central portion of this, we have got the bile canaliculus, which drains into the central vein. The bile canaliculus, which drains into the central vein. We have got, so this is the bile canaliculus. This green area is the bile canaliculus. This green area is the bile canaliculus, which is draining separately, and we have got I'll draw it with a blue color. Yes, somewhere here we have got the central vein, the portal vein which is draining into the central vein. So this is the portal vein draining into the central vein, and I'll label it now. So this is the portal vein draining into the central vein. So somewhere here we have got a round central vein. All right, this is the central vein. And green color ones, so this is the bile ductule. Similarly, we have got here a hepatic artery, and a portal vein. So these are the tributaries of the portal vein, right? So I label it now. This is called as portal triad. So what are the contents of portal triad? We have got a portal vein, we have got a hepatic artery and we have got a bile duct. All right, bile duct. We have got a bile duct, hepatic artery. Oh, I'm sorry, I was blocking the portal vein labeling. Yeah. So I've got the portal, the portal triad. The portal triad consists of hepatic artery, portal vein, and bile duct. Now, if you see the arrangement of these cells in the form of a hepatic lobule, it's something like this. All right, each of this lobule will have a central vein like this here. So this is the central vein, and these are the cells arranged like this. All right, plate-like cells arranged like this. Which are draining into. Now these are the portal triads. I'll show you. These are the portal triads. Consists of hepatic artery. So these are the portal triads, consisting of the hepatic artery, the bile duct, and the portal vein. Portal vein, right? So if you draw a liver like this, you try to understand the liver and draw it like this. So these are the portal triad. This is the central vein. Portal triad and central vein. So if you try to understand this with the help of this diagram, so suppose this is the liver. So into the liver we have got the portal vein entering like this, and separately we have got the inferior vena cava. Inferior vena cava, which is drained by the hepatic veins. All right. Then separately we have got what is known as the cystic duct and the gallbladder. Cystic duct and the gallbladder. So this is the portal vein. All right. This is the cystic duct or the gallbladder, which divides to form the bile ducts, and this is the hepatic vein, which is draining into the inferior vena cava. Hepatic vein which is draining into the inferior vena cava, right? If you take cut section of the liver, it is showing like this. If you take cut section of the liver, it is showing like this. If you take cut section of the liver, we have got hepatic lobules, central vein, portal triad separately. All right, this is the anatomical unit of the liver. We call it morphological unit or anatomical unit. There is physiological unit of the liver. I'll show you what is physiological unit. So from this area, from two central veins like this, a rhomboid structure. So this area is called as hepatic acinus. From one central vein to the other, we are calling it hepatic acinus. We call it hepatic acinus. This hepatic acinus is the physiological unit of the liver. So I'll draw acinus separately. Physiological unit. So we've got two central veins of the adjacent hepatic lobules. We've got two portal triads which contain hepatic artery, which is very, very important. Portal triad which is containing hepatic artery, and this area forms a hepatic acinus. Hepatic acinus, right? This is the physiological unit of the liver. In medicine, when I'm teaching jaundice chapter, I'll again revise this point of physiological unit separately for you. So this is all about the development of the liver. This is all about the development of the liver. Liver develops from the hepatic bud, which divides into pars hepatica and pars cystica, and this is how all these structures are formed. All right. Then the development of bile duct is very, very simple. Actually, bile duct is formed from the pars cystica. The bile duct, right and left bile ducts will join to form cystic duct, and cystic duct joins to form a gallbladder. Gallbladder parts and all we'll discuss in anatomy of the gallbladder and histology of the gallbladder. Right now in the development human embryology, this point is more than enough, that gallbladder is formed from the cystic duct, right? Sorry, pars cystica. Pars cystica. Now let us discuss an important aspect, the next part of today's class, that is the development of the pancreas. Development of the pancreas. I told you that functionally the pancreas has got both exocrine and endocrine functions. So microscopically there are two components of the pancreas. There are two structural components and two developmental components. So everything comes 2 in pancreas. There are two functional components, exocrine and endocrine. Two structural components, that is the dorsal pancreatic bud and the ventral pancreatic bud. So we have got dorsal pancreatic bud. Dorsal pancreatic bud and the ventral pancreatic bud. The dorsal pancreatic bud, which gives rise to, it is first to appear in the 4th week of intrauterine life. It is larger than the ventral bud. It is cephalic to the ventral bud, and it is in between the two layers of the dorsal mesentery of the duodenum. These are the important points. It is in between the two layers of the dorsal mesentery of the duodenum. Ventral pancreatic bud is in close relation to the hepatic bud. Ventral pancreatic bud lies inferior to the duodenum, inferior to the hepatic bud. The ventral pancreatic bud grows into two layers of the ventral mesentery of the duodenum. So dorsal pancreatic bud growing from the dorsal mesentery of the duodenum, ventral pancreatic bud growing from the ventral mesentery of the duodenum. So what happens is, during the rotation, yesterday I told you 3 steps of rotation: stage 1 90 degrees, stage 2 90 degrees, stage 3 90 degrees. So during the rotation what happens is, both the, say suppose this is the duodenum, I'll draw it like this. So this is the duodenum, first part, second part and third part, and then we have got fourth part of duodenum. Alright, so we have got a dorsal pancreatic bud here and a ventral pancreatic bud here. So what happens is, as the rotation of the gut is occurring, what happens is the dorsal pancreatic bud, so ventral pancreatic bud, I'll draw it in some other color. Alright, ventral pancreatic bud. So what happens is, the dorsal and ventral pancreatic bud, they undergo fusion. The pancreatic tissue from the ventral and dorsal bud undergo fusion exactly at the 7th week of intrauterine life to form one single mass. So as the duodenal loop is undergoing shift or rotation, the ventral pancreatic bud along with the primitive bile duct comes to the right side, and dorsal pancreatic bud goes to the left side of the duodenum. Alright, the ventral pancreatic bud comes to the right side and the dorsal pancreatic bud is going to the left side of the duodenum. So this is the duodenum, right? So what happens, there is fusion in the 7th week of intrauterine life, and ventral pancreatic bud forms the lower part and head of the pancreas, whereas the dorsal pancreatic bud forms the rest of the body and tail of the pancreas. Right? So the ventral, so this is the head and uncinate process of the pancreas formed by the ventral pancreatic bud. Dorsal pancreatic bud is forming the body and the tail of the pancreas. So body and the tail of the pancreas formed by the dorsal pancreatic bud. So body and the tail of the pancreas formed by the dorsal pancreatic bud. Body and the tail of the pancreas formed by the dorsal pancreatic bud. Alright, whereas the head and uncinate process of the pancreas is formed by the ventral pancreatic bud. So these pancreatic buds, they have got duct system. Initially what happens, the duct of the dorsal pancreatic duct, the duct system of each of these buds are separately open into the duodenum. So opening of the dorsal pancreatic duct is 2 centimeters proximal to the ventral pancreatic duct. They are by 2 centimeters, they are different. The ventral pancreatic bud and the bile duct, they have a common opening in the duodenum, and we call it the common duodenal papilla, the major duodenal papilla we call it. Whereas the main pancreatic duct, which is formed by the distal portion of the dorsal pancreatic bud, it is it crosses in the midline, and it in its the proximal part of this duct, it opens into the ventral bud. The main pancreatic bud opens into the major duodenal papilla. An accessory pancreatic bud, which is formed by the dorsal pancreatic duct proximal portion, it opens into the minor duodenal papilla. It's something like this. So there are 2 ducts, is it not? So we have got a dorsal bud and a ventral bud. So dorsal duct and a ventral duct. So say suppose this is the duct of the ventral bud. We are calling it as a ventral duct. Alright. And say suppose this is the dorsal duct. And say suppose this is the duodenum. Second part of the duodenum. So what's happening here is, the ventral duct has got a small opening. The dorsal duct has got a small opening into the duodenum. So who joins the ventral duct? The bile duct, which is growing along with the ventral bud. I told you why, when the ventral bud is close to the hepatic bud, because bile duct is growing close, and it comes here and opens along with the opening of the ventral duct. So initially the ventral duct and the dorsal duct, initially it is in line with the dorsal duct. So next what happens is, the ventral bud forms a union with the dorsal bud next. So this is the new union that is formed. This part is a new part that is formed. Alright. So that this duct drains from the distal portion, it is draining from the dorsal duct. Alright. An arrow mark direction is like this. So draining is occurring like this. Alright. Whereas what happens to this portion? This portion remains. It becomes smaller, and it becomes the accessory pancreatic duct. So when you draw it again, it looks like this. So we have got major duodenal papilla and minor duodenal papilla. We have got the ventral pancreatic duct, which has formed a new communication with the dorsal duct, and dorsal duct distal portion is entirely draining like this. Alright. And we have got the bile duct, which is coming and uniting with the ventral pancreatic duct to form the combined drainage into the second part of the duodenum, major duodenal papilla. Now what happens is, this portion becomes rudimentary or becomes accessory. It becomes minor, and it is opening into the minor duodenal papilla. So this was actually contributed by the dorsal duct, but it has now become accessory pancreatic duct. Whereas this area has become the main pancreatic duct. this area has become the main pancreatic duct. So the portion of the duodenum where accessory pancreatic duct enters is called as minor duodenal papilla. The portion of the duodenum where the main pancreatic duct enters, it is called as major duodenal papilla. Major duodenal papilla. Major duodenal papilla. So this is the formation of duct system. All right. This is the formation of the duct system. You will have to remember, it's a very, very important clinical correlation. It has a lot of points you understand in surgery, chapters of pancreas, the pancreatic calculus, the acute pancreatitis, chronic pancreatitis. You will understand it better if you understand this embryological aspect. So we have completed the understanding of pancreas. I request you to go back to the video and again revise it to yourself by replaying the video multiple times. Next we'll discuss spleen. The spleen is formed by small spicules or small portions of the spleen, small lobular portions of the small parts of the spleen. We call it spicules or spleniculi. Spleniculi. Multiple spleniculi. These are nothing but small lobular masses of splenic tissue. Small lobular masses of the splenic tissue. These lobules later on, so small lobules we have, which later on fuse with each other to form a major spleen, which fuse to form a major, a larger spleen. So what happens is, the spleen has got two surfaces. In this surface, one borders, anterior and posterior border, something like that, to which the tail of the pancreas is attached. So here we have got splenic notch. This splenic notch is in the upper border of the adult spleen. It indicates the lobulated development of the spleen. This splenic notch in the spleen is clinically, you can palpate. If the patient has got splenomegaly, you can palpate this splenic notch. This splenic notch tells you that the spleen is formed by small lobules, union of small spleniculi. Union of small spleniculi. Alright? And it is present on the left side of the abdomen, just in the left hypogastrium area, we call it. Left hypogastrium area. Right? Next we'll discuss the formation of diaphragm, diaphragm is a dome-shaped muscle. It's a dome-shaped muscle that separates the thoracic cavity from the abdominal cavity. It is a major muscle of respiration. The components of diaphragm are central tendon, the central tendon, we have got the right crus of the diaphragm, we have got the left crus of diaphragm, we have got a costal part which is a peripheral part, and in the posterior lateral aspect we have got the costal part or the peripheral part. So these are the parts of the diaphragm. Where are these parts developed from? The central tendon is derived from the septum transversum. septum transversum. The right and left crusts are derived from the dorsal mesogastrium or dorsal mesentery of the esophagus. Dorsal mesentery of the esophagus gives rise to right and left crust. Whereas the costal parts, the peripheral parts, are the growth of the muscle that is mesodermal origin from the external body wall. That is called as the pleuroperitoneal membrane. The mesoderm, the muscular ingrowths from the pleuroperitoneal membrane gives rise to these parts. So septum transversum is the most important part. Please remember, the septum transversum is the most important component that contributes to the anterior and median portion of the central tendon. The central tendon is a major part of the diaphragm. The central tendon has got a opening. It has got an opening for esophagus. It has got an opening for blood vessels like aorta and inferior vena cava. So this growth of esophagus, growth of inferior vena cava, growth of aorta is occurring along with the central tendon. Liver also develops, small part of the liver is also contributed by caudal part of the septum transversum. The pleuroperitoneal membranes, on the other hand, which are present posteriorly and laterally, they are small pairs of membrane, the paired membranes, which gives rise to the costal portion of the diaphragm. The mesoderm of the body wall and the external body wall, it gives rise to the pleuroperitoneal membrane, which gives rise to the costal part of the diaphragm. Costal part of the diaphragm. Alright. So the diaphragm, from the 4th week of intrauterine life, the septum transversum is growing from the somites C3, C4 and C5. Please remember this part. C3. So any irritation of this area will result in hiccups, will result in chest pain. C3, C4 areas may present to you with cardiac chest pain or atypical cardiac chest pain sometimes. And this extends as epigastric pain sometimes. Epigastric pain, especially the inferior wall myocardial infarction, which is in close proximity to the central tendon, it presents with epigastric pain, and you may confuse it with the simple gastritis, and patient is actually suffering from a major heart attack, right? So this occurs because of the fibrous pericardium, which grows along with the membrane, that is the pleuroperitoneal membrane, is giving rise to fibrous pericardium. There are a lot of sensory nerve supply to the central portion of the diaphragm, which is pain sensitive in nature. Pain sensitive in nature, and elongation of diaphragm occurs along with the growth of the overlying lung tissue and underlying liver. Overlying lung tissue and underlying liver, there is elongation of the diaphragm occurring, and this gives rise to a major portion of the diaphragm, right? This gives rise to the major portion of the diaphragm. Now, let us discuss the next point. Let us discuss is, we will discuss how the respiratory system is formed. The respiratory system is very unique in its formation. It has got certain subdivisions. It has got certain anatomical subdivisions. Anatomically, it is divided into right and left upper respiratory tract and right and left lower respiratory tract. The upper respiratory tract consists of, if you talk about the upper respiratory tract, consists of the nose, from the nose up to the pharynx, the larynx, all these are included under upper respiratory tract, whereas lower respiratory tract consists of the trachea. The lower respiratory system consists of the trachea, the bronchi, the bronchioles, the terminal bronchioles, the respiratory bronchioles, and the alveoli. All these parts. Functionally, if you see, so we have got terminal bronchioles, we have got alveoli, All these are parts of lower respiratory tract. Whereas functionally, if you see, this is anatomical division. All right. Functional division, if you see, functionally from nose till the bronchioles, it is the conducting part. The upper respiratory tract and lower respiratory tract up to the terminal bronchiole is conduction pathway, we call it. Only it conducts the air. Whereas actual respiration starts with the respiratory bronchioles and the alveoli. So we call it respiration. Actual respiration or exchange of gas. Exchange of gases occurs in the alveolus and the respiratory bronchiole. The development of upper respiratory tract up to the larynx we have discussed in our previous class. Now let us try to understand the development of the rest of the respiratory system. So please remember that respiratory system is purely endodermal in origin. The larynx, the trachea, bronchi, they are having internal linings. These internal linings are lined by the endodermal in origin. This respiratory system has a unique cartilage which supports it, and it makes it highly elastic in nature, especially to conduct the large amount of air in and out. Large amount of air to be conducted in and out. So all these are important functions of the respiratory system. The respiratory system is derived from the respiratory diverticulum. The respiratory diverticulum arises from the growing foregut. I told you that what happens is there is a laryngotracheal tube formed. I'll show you with the help of diagrams, which gets separated first. So we have got a small laryngeal. So this is the tracheo-esophageal fold with the development of the esophagus leg. So this is, say suppose this is the developing esophagus. The developing esophagus on the dorsal aspect, it is giving rise to a small fold like this. We call it the tracheo-esophageal fold. This tracheo-esophageal fold, if you take a cross section here, it looks something like this. So we have got the esophagus like this. This is the esophagus, alright, which is giving rise to a small outpouching called as the tracheo-esophageal fold like this. So the blue color, I'll label it as tracheo-esophageal fold. Alright, tracheo-esophageal fold. Now what happens is, here forms what is known as a small diverticulum develops. We call it laryngotracheal groove. So in this, what happens is it forms ventrally, it forms a groove called as laryngo. Now it grows and forms a groove like this. Alright, to form what is known as a laryngotracheal groove. This laryngotracheal groove deepens. It deepens to form a small membrane which separates the pharynx from the larynx, or separates the upper end of the esophagus from the larynx. The pharynx and larynx are separated. So this groove now, it appears something like this. So this is the larynx and underlying gut like this. Alright, it grows like this. So we have got laryngotracheal tube like this. This is the laryngotracheal tube. The blue color one. And this is the growing pharynx. Alright, growing of the pharynx. Now what happens is, if you take this again, so this was a cross section, is it not? It looks something like this. So we have got the pharynx and the esophagus growing like this, and separation has already occurred. Somewhere here the laryngotracheal tube forms. Now trachea divides into two branches and gives rise to respiratory buds, small respiratory buds, the right and left respiratory buds, which grow into the right and left respiratory lungs, right and left lungs. So respiratory primordium we call it. So initially the respiratory primordium is formed, right and left respiratory buds are formed. These respiratory buds grow into right and left lungs. Alright, so here somewhere we have got the larynx. Here somewhere we have got the trachea. And these are the These are the respiratory buds. All right. The right and left respiratory buds. The right and left respiratory buds, which are going to giving rise to the right and left lungs respectively. So with this regard, we have completed the understanding of the development of larynx, the development of trachea, the development of right and left respiratory system. Then what happens is these buds are going to grow, that they are going to divide into multiple branches. Each of these branch divided into segmental bronchi, then into bronchopulmonary segments. Each lung has got 10 bronchopulmonary segments. 10 bronchopulmonary segments we call it. This I'll teach you in anatomy along with few diagrams. If I teach you, you will understand how the lung is divided into each lung is divided into the 10 bronchopulmonary segments. We'll try to discuss in detail when I am discussing the anatomy, right? Sometimes here during the formation of the larynx and the trachea and the esophagus, if the division does not occur properly, it develops into what is known as tracheoesophageal fistula, something like this. So esophagus is communicating with the trachea. We have got the lungs like this. So we call it tracheoesophageal fistula. There are different types of tracheoesophageal fistulas based on the presentation, which is a common type and what is a treatment and what is a presentation. I'll discuss that when I'll be discussing surgery and pathology chapters, right? So to summarize, the foregut has got a dorsal component and a ventral component. The ventral component of the foregut is giving rise to laryngotracheal diverticulum. The laryngotracheal diverticulum or a laryngotracheal fold, tracheoesophageal fold, or it is growing into a laryngotracheal groove, which is getting separated. We have got separate trachea formed, separate larynx formed. The larynx is divided into thyroid cartilage, cricoid cartilage, arytenoid cartilage along with the muscles. The trachea is divided into right and left principal branches. Apart from that, we have got primary bronchus dividing into secondary bronchi dividing into segmental bronchi dividing into terminal bronchi dividing into respiratory bronchioles, terminal bronchioles dividing into respiratory bronchioles dividing into alveoli. These are the respiratory exchange, gaseous exchange systems of the respiratory system, right? This completes the understanding of today's class. In the next class, we will be discussing cardiovascular system development. If you have any doubt in today's class, feel free to ask me. Peritoneal cavity, pericardial cavity and pleural cavity. We did not discuss it completely because it's not that important for your exams. Please remember that they are developed from the intraembryonic coelom, and how intraembryonic coelom is formed we have already discussed in our previous class. So you have any doubt, feel free to ask me. Till then, have a nice day. Bye.